All right, thank you for the introduction. Sorry, I jumped in a little earlier. I was maybe miscued. Yeah, as I heard today, we'll be talking about using deep geometric and some topological methods specifically for learning and characterizing dynamics from different sorts of cellular data today. Generally speaking, we have a lot of different data types, but it seems like the more dimensions and observations we have in the data, sometimes the less we can get of the true dynamic, time varying nature of these entities, whether they be cells, molecules, or brain activity. But it all kind of comes down to the fact that cells are very dynamic entities that are undergoing a lot of processes constantly. And if we really want to understand cellular systems from this big data, some of the challenges are that we have to learn what's happening in sort of continuous time in order to be able to, for example, disease, which is an example I'll give you later, is an aberrant dynamic that starts to unfold. And if you'd like to stop it, then you have to understand something about the dynamics. And not only just understanding them, the dynamics are operating in a very high dimensional complex space by way of interactions that are constantly going on. So it's even difficult for humans sometimes to see, and you'll see another example later, what the difference between sort of random dynamics or very regulated dynamics are. So there's a problem of also characterizing them. So just generally speaking, when you have high throughput, high dimensional data like we do in the biomedical sciences, like the examples I showed with single cell RNA sequencing or ATAC-seq or some of these other modalities, there have been a lot of challenges as far as denoising the data, distilling the state space, integrating multiple modalities, and also this idea of learning back static snapshots. And we and others in the field have been working pretty hard since the advent of these data types around 2015 to tackle these challenges using various methods. But today I'll be talking about the idea of characterizing dynamics. So in single cell RNA sequencing data, we have the issue that we don't actually have rich time courses. We may have 4 or 5 time points at most from any process that's taking place, or you might see a pseudo-time dynamic that just arises from the heterogeneity in the sample. But what we'd like to understand is how is the cellular population evolving? So if we have a discrete time course, what would have happened at a time point that we didn't measure? And also, what would the fate of a cell been if we had been able to follow it? Instead, we all know that single cell RNA sequencing kills cells and you can no longer follow them. So these are some of the ways we'd like to infer dynamics from single cell data sets. Once it's inferred, you might see that the dynamics actually takes very complex patterns. This is a newer method from my lab called temporal fate, which is showing the dynamic activation pattern of neurons. And you see that these dynamics are fairly complex. So how can we compare different dynamic regimes and what's really different about them? So that's what I'll try to talk about towards the end of the talk. So, just like the way we tackled some of the other earlier challenges like denoising and data visualization, it turns out that Riemannian geometry and specifically data geometry really helps us solve these challenges. The idea is that there is some regular structure, like a manifold, that you can model the data as arising from. So you've sampled from an underlying lower dimensional manifold with noise, and this turns out to repeatedly be a successful model in terms of the output downstream. The reason this is, of course, a good model for scientific data like biomedical data is we're measuring a lot of different features, and these features are all pertaining to the same entity. So these features are all somehow ultimately coordinated. So they have to lie in a restricted lower dimensional space. And this restricted lower dimensional space gives it this lower dimensionality of the manifold. And really, the sheer number of points that we can sample makes it so that the manifold shows all the viable parts of the state space in a smooth way. One thing I should mention is when people talk about the manifold assumption, the methods we use are automatically applicable to collections of manifolds. So it doesn't have to be a very single one, because the methods we use to understand and learn manifolds are generally graph-based methods. And so the graphs naturally model like internal affinity between different manifolds. So when we say manifold assumption, we really mean collection of manifolds assumption. So how do you learn these internal lower dimensional structures of cellular data? And as I hinted at, we try to render this point cloud data as a graph. So when I say point cloud data, you can think of each cell as a high dimensional vector living in some high dimensional space, and it's sort of creating a cloud of points. But by modeling it as a graph, you start to uncover its lower dimensional, quite nicer structure. So the simplest method for turning point cloud data into this kind of graph is to go from an observation by feature matrix to a distance matrix. And for this, typically in cellular data, you can just use Euclidean distance or some simple distance like that. And for example, the distance can be swapped out to, if you want, for example, cosine similarity, cosine dissimilarity, or whatever choice you have for that. And then you can go from distances to affinities by way of some kind of kernel function. The really important thing about the kernel function is that it have a drop-off like this. So it's emphasizing the close neighbors. Of course, if you turn this into a box-shaped kernel, you'd get a k nearest neighbors type of graph where things are connected only to their close neighbors, but by radius. So another additional step in learning the manifold that turns out to be important is to turn this affinity matrix that we get going from cells by genes to cell by cell distances, then to affinities, is to Markov normalize the affinities. And there's several ways to do it. Some ways divide out the effective density. And what that gives you is a transition operator that conducts random walks on the graph, and that turns out to be very important. If you raise this matrix to the power of t, then that's as if you took a t-step random walk. And if you right multiply it by a vector, it means you started the walk there. If you left multiplied it, you'd sort of get the how points or signals would collect at the end of the random walk. And so you can create a diffusion operator out of any graph, actually. You just take the adjacency matrix of the graph and Markov normalize it like this, divide out by degree to the left, and you would get this kind of diffusion operator, which turns out to be hugely useful. The reason the diffusion operators are useful, as well as graph Laplacians, is that they're actually discretizations of heat. And it turns out heat diffusion on a manifold is an interesting way of figuring out manifold intrinsic distances. And this was even known in the 1960s in the Varadhan's formula setup, where log of the heat diffusion operator directly gives you manifold distances. And this has been shown many times. This is one of the reasons why, in addition to just the graph, diffusions on the graph will give you very good ideas about distances on the manifold. The first time this was exploited was in the 2006 paper by Kaufman and Lafon on diffusion maps. So diffusion maps perform an eigendecomposition of this diffusion operator and weight the eigenvectors by the eigenvalues. And this creates a new latent space where Euclidean distances have some connection to manifold distances. However, we don't often use the whole diffusion maps construct. We use the diffusion operator because it has a lot of interesting information intrinsically. Diffusion maps, for example, tend to disambiguate the dimensions or the paths in the data. So all of this would be highlighted by one of the diffusion eigencomponents, and this would be by another, and which is why spectral clustering could pick these out, whereas clustering in the ambient space might not. And so one of the ways we use this kind of idea is to create a latent space that preserves this diffusion affinity information. And we perform operations like machine learning operations like clustering in such a latent space. And I'm going to show you examples of how we do that in order to learn dynamics. And finally, one thing of note is that the eigenvectors of the diffusion operator actually form harmonics in the graph Fourier space. And these graph Fourier harmonics can be useful for denoising and removing variance from signal. And this we had used previously in our denoising methods like magic, but more recently in our topological methods like diffusion condensation. So these ideas we use to tackle some of the challenges that I told you about before, denoising, visualization, comparing multiple samples or data sets. But today I'll be talking about utilizing diffusion geometry as well as ideas from graph topology together with deep neural networks in order to learn dynamics. So the first concept that I'll talk to you about that will allow us to learn dynamics is the concept of optimal transport. So you saw before that what we had were point clouds of cells. And if you have multiple samples like these, like if these were time points, then you might want to transport one to another in order to learn continuous flow in latent space. Initially, we had a method called trajectory net that did this, but it did it sort of in a Euclidean type space. But more recently, we have a method called MioFlow that I'll talk about that uses optimal transport on this data graph space in order to model it within the manifold. So before I get there, I'm going to briefly talk about a method of how do you compute optimal transport that we developed in 2021 for ICML. The main idea is to use multi-scale graph density estimates to define an Earth mover's distance or an optimal transport distance. So Earth mover's distance is the idea that there's work that could be done to move this pile of dirt to this pile of dirt. And if you have an optimal way of moving this, then this actually provides a distance between these two probability distributions. And so an optimal transport plan is actually a joint probability distribution or a coupling that tells you how to move the dirt from here to here so that the cost of moving, which is proportional to a ground distance, is minimal. And this is usually solved by a linear program. But if we have huge point clouds of data, and particularly tons of data points as you would get with patients or some other high throughput modality, then you might not necessarily want to solve these linear programs. They get very expensive. So an alternative way is to find a witness function that exposes the difference between these two distributions, with the caveat that the witness function is one Lipschitz so that it doesn't suddenly jump up in certain areas, sort of cheating. And it turns out that the Kantorovich-Rubinstein duality ensures that the expectation of these probability distributions subtracted under these two distributions gives you the same as the optimal transport plan. So now this can be very useful in creating a very fast method for Earth mover's distance. So this kind of idea was used for distances between images back in 2008, where each image was modeled as a histogram. There was a difference histogram taken. It was decomposed in terms of bounded functions, these Haar wavelets. And then the Earth mover's distance was computed as a weighted sum of the difference in these weighted scales. So we wanted to do something very similar, but not on an image, but on an irregular graph structure. So exactly the graph structure we created from the point cloud cellular data. So earlier we had discovered an efficient method for creating a kernel density estimate on graphs. So the idea is that we would combine our point clouds together, create indicator signals for each of the samples, and then you basically smooth these to get a kernel density estimate. You can actually derive this as a solution to this optimization. But what we discovered is that you get better results when you change this optimization slightly to create a heat kernel there. And so when you do that, you get these density estimates, and we turn the density estimates into likelihoods, which tells us where in the cellular manifold each of the samples is likely to get signal from. So if you take these T cells under control and stimulated experimental conditions, you see that the highly likely cells under the stimulation are here. So this means these are the most activated cells in the population, even though I'm combining both the samples here. So before we'd use this for comparing just a couple of pairs. And once this method was completed, we could do hundreds of samples and optimal transport between hundreds of samples. So we repeat the same trick. We take all these point clouds of data, turn them into a joint data graph, and then we create density estimates by creating indicator signals and then diffusing them to different scales to create different scales of kernel density estimates. And these are very similar to the different scales of wavelets that were used in the wavelet-based earth mover's distance. And then it turns out you can just take an L1 norm to get an earth mover's distance here. And what you see here is that this is a multi-scale diffusion distance. So these indicator vectors, they're all diffused to different scales, and you have differences between two different scales of diffusion, which is called a diffusion wavelet. And so this is very literal translation of that kind of method. And we were able to prove that if you compute earth mover's distance using this, that you would use a manifold ground distance. And the proof relies on the discretization of the current heat kernel using the diffusion operator, which I showed before. What we were able to see is that it's very accurate, but it's much faster than the other methods that we have. And this is another one of our own methods that was more recently published that's a primal method. It's called geodesic sinkhorn. And what you see here is that if you had you had little Gaussian distributions around this moon-like shape, that the diffusion earth mover's distance actually gives you the correct optimal transport between them. So each dot here is a little Gaussian distribution. It's centered at a certain point in like a semicircular shape, and you get the right configuration of them. And we saw that with COVID patient data as well. Each of these dots is a COVID patient. There was a point cloud of single cells collected on them. And when you embed them based on their optimal diffusion EMD, earth movers distances, you get the right configuration of the patients, where the patients who died are over here, and they also have low T cell counts. They're over here. They have low T cell counts, and these have low counts, and they're the ones who would have died. So I said that backwards initially. So we use diffusion earth mover's distance to create this kind of Wasserstein embedding, where between every pair of data points, you have an earth mover's distance. But back to our original goal, can we use them in order to transport cellular population from one to the next in order to define a continuous dynamic from these static snapshot data sets? So this was published in NeurIPS 2022. The main idea is, of course, to embed into the multi-scale diffusion distance space that I showed you before. So basically a space where distances are this kind of distance, and then run a neural ODE in that space and penalize by optimal transport to match. And so the idea is we had 4 or 5 time points of single cell data. There are other methods that are complementary to ours, but they do different things. So single cell RNA velocity gives you instantaneous arrows, not the entire flow. There's methods that do pairwise sort of matching, but rather than a continuous dynamic inference. This is our earlier method called trajectory net. So here we create what's called a geodesic autoencoder. And in that autoencoder, distances between data points is a multi-scale diffusion distance using such dyadic scales, 2 to the power of something. And so once you embed data in there, you are sort of straightening the manifold distance into Euclidean distance in that space. And then you can run a neural ODE, which by now a lot of people are familiar with. These are neural networks that learn dynamics, but we're doing it in a latent space. That means that the neural network actually learns the derivative of each cell at each point, and then you have to integrate it out to get the next position at the next time point. And this is done very fast using this adjoint sensitivity trick, which I won't go into. It just allows you to skip back propagating through the ODE solver. Okay, so the neural network gets the derivative of each cell, you integrate it out, and you get the new time point. And at this new time point, once you've transported all the cells to that time point, you can penalize it by your known population at that later time point using normal earth mover's distance. But because you're in this latent space, you get manifold earth mover's distance as the penalty. So we can impute continuous trajectories using this. We also noticed that if you penalize the magnitude of the derivative, so you try to keep these derivatives to be as small as possible, then you get these smoother paths. And these smoother paths seem more realistic. They're wasting less energy. And it turns out that that gives us a construct that has been recognized before, and it's called dynamic optimal transport. So dynamic optimal transport is a type of optimal transport that's also penalizing the actual paths rather than the end result. And we are able to show in the paper that so if you have this energy penalty, then you get dynamic earth movers, dynamic optimal transport with a geodesic manifold distance as the ground distance between data points. So the idea of adding this energy regularization is that you minimize the energy you take to perform the optimal transport. So things go right into the spot they're supposed to go when they're transitioning from one population to another population. The good thing about neural networks and why we tend to put these things into neural networks rather than just sort of computing them analytically is because there's a lot of different things going on. As I show, there's a lot of different processes in cells. They're not just transitioning, they're making decisions about dying, they're making decisions about proliferating. And so you can add those as other regularizations, and you can tune the proliferation rate, for example, using other estimates of how much the cell might be proliferating based on its gene expression profile. So in order to add cell death and growth, we can allow imbalanced transport. And we can actually continuously learn a growth rate for each cell, which turns the system into a PDE, but we've actually shown that we can do that. And if you have complementary measurements like RNA velocity, you can regularize to those. You're penalizing to match the RNA velocity that you get. And so we showed that using this whole framework, we can take something that's pretty complicated and tracing out curvy paths in space pretty successfully. So going from these purple to blue to green to yellow, and you can see that we don't have a lot of lines cutting through the middle. That means that the paths, as much as possible, are following the density of the data in a way that even our earlier method didn't do. And this is Schrodinger's bridge has difficulty doing. So this is really ideally suited for transport rather than some of the flow-based methods that are, if you go to an ML conference like ICML, which is going on this week, a lot of flow-based methods are used for data generation, but rather we're using them to really understand dynamics. So shown on the earlier data set, you can transport population of cells, and in the wake of the transport, you draw out these trails, and these trails give you single cell trajectories. So you can see that this is different than pseudotime because it's giving you a trajectory for each individual cell, and you can see these cells diverging. We've been using it recently, and we have a preprint out, which you can check out if you want, on trying to understand dynamics in triple negative breast cancer. So the dynamic we're trying to understand is how can primary tumor cells, which sometimes aren't very aggressive, how do they actually transition to a more aggressive state that could create a metastatic tumor? And we're studying both the process of epithelial to mesenchymal transition, as well as the reverse, mesenchymal to epithelial, which are both needed for metastatic transitions. Turns out EMT is better studied than MET. So together with our cancer collaborator, Christine Schaefer in Australia, we grew these breast cancer cell line cells into these mammospheres, which are like breast cancer organoids. And you see that some of them grow very big, similar to a secondary tumor. And we have, again, collected 5 time points of data, day 0, 1, 12, 18, and 30. And they are arranged like this. So you see the transport would roughly be like that. And so you can transport the cells, and you see that some of the cells transition to form these bigger tumors. Other cells go towards this apoptotic state where they die. And what we're really looking for is what are the cells that transition. The unbalanced transport allows not all the cells to transition. A lot of them will die under this condition. So one thing we can do is called cell of origin analysis. So we can specify the cell cycle stage of the cells that survive, G2M, and we can finally characterize it with using many gene markers, which we've validated experimentally together with our partner. The other thing you can do with these kinds of single cell trajectories is you can group the trajectories. There's these trajectories that are going to a metastatic type state, and these are the ones that are going to a benign apoptotic state. So what is really different about these? And it actually turns out, if you zoom in on here, there's some that go to a very metastatic state that won't allow them to revert, and some that go to a more sort of epithelial or mesenchymal state versus epithelial state. So these kinds of trajectories, we can compare them. You could do time-lapse imaging, but as I mentioned, this will give you one dimension of information. So you could validate one or two or however many that you want a time-lapse image in order to verify this. But the interesting thing is when we get these types of cellular trajectories, we also get gene trajectories that come along. So these are gene trajectories. These are genes that are on first, second, third in this set of cellular trajectories. And so we can create rich gene networks that are based on time-varying measures of causality, here using Granger causality. In order to do this, more recently, I'll explain another method we're using, but this network was derived by taking these gene trajectories and running Granger causality, and that gives you this network of genes, and they're organized by time. So gene cluster 2 is generally turned on before gene cluster 3, and you start to see how this network sort of cascades to create these gene trajectories. And our hypothesis is that some of the hubs in these kind of causal gene networks might be usable as therapeutic targets. We know at least this one, AR, androgen receptor, is already under clinical trial. We're currently looking at this subnetwork to find additional targets like these. And we have some idea that knocking these out might help the EMT process. So for example, when you knock out this one right here that's at the very top, you get a resurgence of ecodierin, and you get a diminishing of ZEB, which is a mesenchymal marker. So it's perturbing the EMT process somewhere. So that's where we're at with studying cancer dynamics with that. Meanwhile, we developed another neural network because we kind of found it awkward to create those gene and cellular trajectories separately from the network inference. So we thought, what if we just combine both the methods together? And this is a method that's on arXiv. It's called Routini, and it stands for Regulatory Temporal Interaction Network Inference. And the idea of this network is that it's actually a graph attention network. And so the neural ODE network that MioFlow has that I explained before is replaced with a graph ODE network. So the regulatory graph is like right inside the neural network. Our idea was that when you see a cellular trajectory, what that really means is that all the genes are changing temporally. But these gene changes are a result of changes in expression patterns in response to altered interactions between regulatory components. So can we actually get the neural network to model those generative dynamics that follow these principles? Of course, this is sort of an inverse problem. There could be many graphs that could match the same dynamics. So one thing we do is we have a very good prior educated guess. And you could even use the Granger causality technique that I just mentioned as a prior guess, but you'd refine it by getting that network to predict in a time-varying fashion the gene dynamics of the cell. So the way the architecture works is each of the nodes in this graph is actually a gene or whatever regulatory element you're looking for. We've tested it on both gene regulatory networks and neuronal activation networks. And then the job of the neural net graph attention network is to predict the dynamics of each gene within a single cell. But as it's doing it, it changes its attention weights, and the changes in its attention weights are what we infer the network from. When there's an attention that's strengthened, that means that this gene interaction potentially strengthened. And the same thing for if it weakened. So you actually get a time varying graph out of Rotini using the task of predicting the gene expression dynamics so as to match our inference from MIAFlow, or so as to match the inference from real time lapse data that you have on certain dimensions of this. So the graph, this is technically a graph attention ODE network. So this network is again finding a derivative, but this derivative is parameterized by attention over time and attention over neighbors. So each node here is paying attention to its neighboring nodes, which are other genes, and it's also paying attention to itself. So that gives it the ability to have some time lag here. The attention paid to neighbors is on a slightly earlier expression pattern, and you can model autocorrelation. So the gene has some inertia in how it changes. And these attentions are modeled by networks. And these networks take into account time traces of whatever gene that you're looking at, which allows you actually to even model hysteresis, which we're not sure exists in this setting, but it exists in neuroscience. So the basic summary is that this is an attention network that gives a time varying graph by way of a graph ODE. It can support hysteresis, so it can support looking at an interval of time beforehand to develop this attention. So you're looking at some interval of time up to the interaction between two different genes in order to see how to strengthen or weaken the attention. But the output is still an instantaneous derivative. So this graph attention network outputs the derivative, which is again integrated with the ODE solver. But this derivative, as you see here, has two types of attention over time and attention over neighbors. And the overall loss function for this is that it matches ground truth dynamics. And if you want to put optimal transport in, then you can just run MIAFlow and match its results. It's close to a prior network that you have an educated guess on, and it's sparse. So with these loss functions, we're able to train Rotini. One interesting thing what we found was that if you included mild perturbations in your system, then it can elucidate the network structure. If you give it too large of a perturbation, then it would rewire the original network. So the example is you perturb node E, the effect trickles to node D, and then it would trickle to these. Because of the time lag, it would trickle to them at different points in time, and you can understand how this network is wired that way. So we've had results on different systems. So we've done dynamical system simulations, both in neuronal networks and gene regulatory networks. And of the methods that we compared to, we're currently getting the best results. And the actual output that we're interested in is what the network looks like as the time variance goes on. This is sort of a time average network, and we're comparing that to ground truth. We're doing the same thing when we look at gene regulatory networks. Gene regulatory networks tend to have more of these kind of module-like structures, which we're examining as well. The next frontier sort of this is to incorporate spatial information. Spatial information, we're thinking about modeling it as a general term that encodes something about the microenvironment. So this would make this into a conditional ODE, but you could also directly model cell-cell interactions as well and put those into your graph. So we're sort of experimenting with both these kinds of things right now. And so in the final part of the talk, I want to talk about, okay, what do you do after you have these nice time varying dynamics? One of the things we did with the cellular dynamics was look at those causal gene regulatory networks, because we're interested in disturbing some of these dynamics if they're dynamics that are malicious, like the transition from benign cells to very aggressive cancer cells. But we could also just observe the dynamics to see if there's something aberrant going on. And this is when you have data that's potentially more oriented with cellular signaling. And so we have a recent paper out in the Journal of Cell Biology that's using this, But we could also look at aberrant dynamics in the context of neuronal networks. So I'll show these both examples here. So this idea came from a system that Valentina Greco, who sat next to me at Yale, but now I've moved into a cool new building that maybe some of you can visit sometime. So she had calcium signaling in epithelial cells. So epithelial cells are like the cells I was talking to you about in breast cancer. They're in this kind of grid form, and you can see calcium fluxing between these cells when you measure them with something like 2-photon microscopy. But the question is, calcium signaling hasn't been extensively studied in cells that are not neurons. So what are they doing in epithelial cells? Are these just random flashes? Is there any coordination? Really, what's going on? And how can we tell if a cell is not flashing calcium the same way as other cellular tissues? So in order to really quantify and understand this, we started to build a pipeline for characterizing dynamics. So here we already have the dynamics, we want to characterize them. First, we create an adjacency graph from the physical adjacency. Then we do a bunch of graph signal processing, and I could explain what that is, to understand the pattern of signals at a single time point. And then we can map out the trajectory, how they're changing over time, using a variant of our FATE method. Originally, we did it with FATE. Lately, we've switched to T-FATE. And then you can see these dynamics are kind of looping. And you can actually quantify this looping behavior using topological data analysis. So that's the kind of pipeline we have. The graph creation is pretty simple. We're just creating a graph based on cells that are literally adjacent, because these are epithelial cells. The calcium is modeled as a signal on this cell-cell graph. And for graph signal processing purposes, you like the graph Fourier transform of a signal, which moves it to the graph Fourier space using eigenvectors of either the diffusion operator or the Laplacian. But I'm going to skip this. What we actually do is we use something called a graph wavelet. And what these are differences between different scales of diffusion. And we've seen different scales of diffusion being used before, right? We saw them in the density estimation. So it turns out that you can create wavelets on these kinds of graphs by subtracting two different scales of diffusion. And instead of using a graph Fourier transform, you could use a graph wavelet transform to capture these signal patterns. And just like in regular signal processing, graph wavelet transforms are localized both in the location, which is the vertex here, as well as in their pattern. So they're both have a time and frequency kind of localization ability. So now from there, we create something more complicated called a geometric scattering transform. The idea here is you take many different scales of wavelets, a cascade of wavelets. And so you have a multi-scale wavelet transform. And this multi-scale wavelet transform can be repeated, potentially using different scales to create a deep network of wavelet transforms, which is like a deep neural network that creates a rich representation of the dynamic. So just to go a little bit further into this, you use the diffusion operator I created before, use two different dyadic scales and subtract them, and that'll give you wavelets that are localized somewhere in the cellular sheet space or the physical space also. And then we create a scattering transform here using three different scales of wavelets. After these three scales of wavelets are applied, you can get the scattering coefficients of your signal interproducted with these. And you can also take their different moments or other nonlinearities. And then you can take the results of these and do it again to create sort of a deep neural network of those. And we've also shown that parameters of those can be end-to-end differentiable. We have a method called learnable geometric scattering. These turn out to be very powerful signal descriptors, but also descriptors of features on graphs. Finally, we take the description that we get out of the geometric scattering transform at each time point, and we draw a FATE trajectory out of it. And FATE, as you've seen before, is a method that can use these kinds of, this same kind of diffusion operator and get a distance that's based on the log scale diffusion probability. So again, using Vardhan's formula that captures manifold distances between data points. So these are related to diffusion distances, except for they are a triplet distance, meaning if you want to take the distance between x and y, you go through all points z and you're comparing the diffusion probabilities of those. And so this is again related to Vardhan's formula, which shows that the log of a heat kernel is proportional to manifold distances. Recently, we extended FATE to have a multi-view kernel with also an autocorrelation kernel for modeling data points that are staggered in time, and you can read about that here. But this is what it looks like as the calcium is flashing. You start mapping out this FATE trajectory. Remember, each data point is a descriptor of the spatial signaling pattern. And as the spatial signaling pattern evolves, you're tracing out some kind of trajectory in time. And maybe what you're seeing is this is not random. It's a very directed trajectory. It has very low dimensionality. It's not scattered all over the place. And it seems like these cells have a plan when they're performing this dynamic. So we were surprised to see this. So this seems to be highly, highly coordinated, regulated dynamics. And we're able to capture the very nice looping structures that now went away using a persistence homology. In topological data analysis, we can count the connected components, loops, 3D type looping structures, and these topological characterizations all come from TDA, where you're counting the number of holes in any kind of system. So this is one connected component, one cavity, 0 holes. This is 2 different cycles, still 1 hole, and still 1 connected component. So we're currently just doing this with Viatorus RIPS, which takes each of those data points, advances this kind of epsilon ball, and counts the persistence. So now when you do that, you both get a quantification of this whole dynamic that you can just use to compare these dynamics, but you could also put it into a classifier. So you've taken a way of taking this complex time-varying dynamics and just pushing it through into these persistence features so you can train a classifier off of it. But something interesting to note is that if you studied calcium dynamics in the visual cortex of a neuron, it does not have this kind of sort of very coordinated, smooth, sweeping pattern. It's very much more scattered, and the subsequent activations can sort of jump. The idea is that the visual cortex is probably getting a lot of input from the outside. So whenever it sees something different outside, it's kind of jumping the state. These calcium cells aren't. What they're doing is they're doing some kind of signaling to maintain homeostasis. And because they're doing signaling to maintain homeostasis, they're just kind of sweeping smooth arcs. So it would be kind of like, are you okay? Are you okay? Are you okay? This kind of check that they're doing upon each other. And what we see is if you perturb other homeostatic mechanisms like cell cycle, you would perturb the smoothness of this space. So if, for example, you stalled cells in one of the cell cycle phases, G1, you've disturbed the dynamics that they have. It turns out that the G2 cells are the ones that do a lot of this signaling. And you can do the same thing if you yeah, with the topology. The topology actually gives you a neat way of quantifying it, and you can just measure, again, Wasserstein distance between these different persistence diagrams, and you can see that the ones that are enriched for G2 all act very differently than the ones that are stalled and the ones that are control. The other thing we can do is perturb certain proteins that are part of these kinds of mechanisms. When you do that, you also get another idea of what perturbs the dynamics. So if you perturb CX43, you get kind of choppy dynamics again. And so we're using this to study these kinds of patterns and what perturbs the dynamics, which will give us an understanding of what is actually controlling it. And it turns out it's many, many genes. And when we've stalled the cells in a particular cell cycle, or we've perturbed a particular gene, it's only perturbing one part of it. So there's many components that are going into this kind of high degree of regulation. That's actually pretty apparent when you see the smoothness of the dynamics. So we're looking at another data set of signaling in the mouse epidermis and different drugs and how they perturb that. So how do we test the ability of GSTH to actually make these conclusions? Again, we have to do them with simulated mathematical models. So we have different mathematical models like the Kuramoto model, intercellular calcium signaling models, and we've tried different methods as well as replacing parts of GST8. So we've replaced the persistence homology with an RNN. Turns out it's less powerful. We've ablated fate and tried other methods. It turns out that the geometry preservation of fate is very important for understanding these trajectories. And that's how we're able to design the best pipeline for this, even though it seems complicated, by testing the recovery of parameters you put into these simulation systems. So this is where I kind of wanted to stop and see if anybody had any questions. We have a lot of other sorts of methods in our lab. So the diffusion EMD I talked about, you can download and play with it. Same with T-Fate. And there's projects that I didn't talk about having to do with alignment and things like that, but you can find that all on our GitHub. So yeah, please visit there and let me know if you have any questions. And finally, I really want to thank all of my collaborators. So as you can imagine, I'm not an experimental biologist, and yet I showed you data from cancer, which is collected by Christine Schaefer and her wonderful trainees, Shabarni and Bea. I showed you really awesome calcium signaling data collected mainly by Jess Moore from Valentina Greco's lab. And John Locke, who collected the imaging on the cancer story, as well as our other collaborators, Guy, Wolf, Guillaume, Huguet at Mila, who collaborate more on the computational side, and Erica, who's our neuroscience collaborator. Thank you so much.
Okay, what a whirlwind of methods, Smita, thanks. So any questions? The easiest thing is if you can come to the front and you can have the microphone. While people are thinking, I've got one, Smita, which is, you mentioned when you were talking about the learning these regulatory networks about how small perturbations can be more informative than large perturbations, because I think you said the large perturbations can lead to like a completely different network. And yeah. I was wondering, is that's not just because the problem is a little bit unidentifiable, is it? Or I wasn't quite clear why you would, because I wasn't sure what the generative model for the data was in that example.
I mean, so the main idea is if you completely knock out an important gene, the cell has smart mechanisms to compensate for it. It'll use other genes. It'll start turning on other interactions. So if you want to study what the network is at that moment without perturbing it too much, then a mild perturbation would be better.
So it's just that the model doesn't contain everything about how the cell is regulated, and all those other things have changed because the perturbation is too big and sort of put the cell into a different place somehow, or?
Yeah, that kind of harsh perturbation artificially puts your cell into a different place, so you have other compensation mechanisms. So I suppose you could follow the compensation if you had very fast time courses that we're looking at those, so you could actually model the dynamics of rewiring towards the perturbation. We often don't have those. When people give you a CRISPR data set, it's been perturbed and they measure something 5 days later.
Okay, thanks, Peter. We got a question here.
Hello, thank you for that great talk. Normally, biologic data is rather noisy. And from my first glance at the calcium flashing, I would have probably said, yeah, that's some noise. Still, you found out that there's some underlying, actually very one-dimensional dynamic beneath that. Would you assume that in many other contexts where we assume that we just have a lot of noise, there is actually an underlying dynamic that we did not yet discover? Or is that one specific case where it worked particularly well?
So I don't think you can very easily say what the dimensions of the dynamics are, right? So for example, I showed you this plot with the neuronal data. That's also low dimensional, but it's not, I don't think it's quite one dimensional. It's probably three or four dimensional. So generally speaking, in my experience, biological systems are not stupendously high dimensional in what you're looking at. They're definitely lower dimensional than the ambient measurements we currently have, but it would be difficult to a priori estimate the dimensionality of that if that's what you're asking. But it's not that hard to estimate the dimensionality after you analyze it with sort of a manifold learning or a structural learning method.
Okay, thank you.
One more.
Hi, wonderful talk. So just to clarify each point in that three-dimensional graph where you have the overall signaling sort of representation at an instant, correct me if I'm wrong. So
are you talking about this representation I have on the screen right now?
Yeah.
Okay, so each data point, each point here is the signaling representation of one instant, as you said, and the subsequent points are other time points. So
right, that's an evolution through time.
Yeah,
right?
Right. Exactly.
And like each one of these points is when, you know, when we say representing the overall signaling state of kind of all these cells.
Exactly.
So how does that, or like, where are the invariants in that? Is it in like in the number of connections between how many cells, or like intuitively, how would you, because if you have, for example, like 1,000 cells on there, and they have a specific sort of signaling, let's say that in their current state of signaling, they create some kind of instantaneous infrastructure, you know, just to use an analogy, right? And then that kind of like evolves through time. So yeah, like this. So would there be another configuration which gives you the same point in that three-dimensional space where which you were showing?
No, so the idea of the kind of, okay, so there's a few things that are at play here. So I have to go a little bit into how we're processing each time point. To process each time point, we're using something called the geometric scattering transform. So the geometric scattering transform gives you basically sort of node level representations that are then aggregated into a graph level representation, and I have sort of better pictures of that somewhere. There are some invariances built into that. That dynamic is, for example, permutation invariant. And it's also sort of type frame. That means that it's, if you have sort of a slight perturbation, you have a guarantee that the results of the scattering transform will also only be slightly perturbed. So it's resistant to perturbation. So these invariances that come from the scattering transform are then transferred over.
And that, for example, would be, let's say, that image that you showed with the specific, you know, this like instantaneous state of calcium signaling. Like if you were to just rotate the signaling by, say, 90 degrees, would it be the same point on the, so it would be invariant like that? Look, that would be an invariant.
Yeah, it would be invariant to that. If you rotated this whole tissue, it would be invariant to that.
And have you done any sort of analysis into these invariants, kind of see what those are about, or?
We have a number of works on the geometric scattering transform. The geometric scattering transform was actually invented by our collaborator Guy Wolf, together with another collaborator who has since left academia, Matt Hearn, and Stefan Malat. Actually, they were both postdocs in Stefan Malat's lab when they started inventing that. So I became very interested in that right after that paper came out, circa 2019. And we have a paper called Learnable Geometric Scattering, because what I thought was we should put some of these capabilities into graph neural networks, because graph neural networks can sometimes actually be pretty weak at graph representations, and they don't have these very rich node representations that are multi-scale. So we've also shown that relaxing several parts of the geometric scattering transform and learning them maintains these. We have another paper on doing this same kind of scattering on point cloud graphs. We also have some guarantees there. So I think the main invariances that have been studied in that context are permutation invariances and sort of perturbation invariances.
Great, very interesting. Thank you so much.
Mhm.